Good morning. Good morning. Today we are going to talk about this molecule called DNA, and we're going to talk about genes because this is fundamental to the process of evolution.
Ta thari nganzo dudul DNA laj do di korla shares and di khari chagor lana thanda nganzo philgyur knowledge ta shizaro zo di chadyo res.
DNA is the genetic material for every cell on this planet.
Ta di say kola zamling na ta oina thabung khande ji nei di rigje gyusha di ta oina dudul DNA di chadyo res.
It's shared with us and some very simple organisms like the single-celled organism yeast.
Dila du ngaran so imba inare ne thabung kyangde yeast do chasha inare dinju changmanal kharir lena di gyusha di res DNA di.
Yeast make a new organism every hour and a half, and their genes are about 40% shared with us.
Yeast la di kama shivju na Quran kebel do ni suwe res. Ang Quran so rigze ta oina ngazu ta du oina kya shivju chi thungu res.
Some very simple animals, like a certain kind of worm, their genes are even more like us, like 60 to 70%.
Dila du bu chunju rigs do kangpa mejo rigs yo busi bu dinde rikta oina gi Quran so gi rigze ta oina ngazu da khagas thungu la oina kya thujus thungu res.
As we touched upon before, this is a molecule, a piece of a DNA molecule.
Ta thanda di dudul dita oina di DNA dudul chigres. DNA dudul ki chashe chigres.
It's like two strings winding around themselves in a spiral.
Dila du kubani rosh thong do astela. Kubani di chu de yo ro chu su de chadu res.
This is the backbone that holds one strand next to the other, and it's got certain molecules that are exactly the same all the way up and down. This blue stripe is identical throughout the length of DNA.
Tadi kubash do mombudi di thanda chashe shten dos di ya do ene DNA dudul ta na chazang di kubo di ya ba du du res an di niga pensu chigi chigla ni di chadu res chipa chadu res.
But here in the middle is where the variation occurs.
Tadi chusu kuba ni di chipa res la ra thungu res thana shi di kila chig do themba do di ani khaba yo res dila.
These things that look like steps of a ladder are called bases.
Tadi nganzo themba roju thongu do asti dila an nganzo base la yo res.
We only have 4 of them in DNA.
Ta thanda base la di khajo lo na thanda ni chala phusung le yo di cha de ra di khajo la na DNA ta oina dinde rikshi res.
Their structure is such that when they face each other, this yellow one called cysteine is always across from its buddy here, the G, called guanine.
Oh, ta thanda dela seu ji tang dos di cytosine res di kharida thundi gor la na guanine nyam do thundi ge res.
So green always faces a yellow. Green always faces a yellow. Green always faces a yellow.
Dila du khorani khajo chi la chikta de yo dinde cha de res. Ta dina ta na jangu di jangu tang do di seu tang dos jangu da seu ni khajo phasu ta de yo dinde ju res dina ta oina.
Likewise, this A always faces a T.
Thana shi di ti jik dos di e da khorani pensun ta de yo dinde cha de res.
So here pink is always across from orange all the way through.
Dila du di singya che di liwang sungu do dina ta oina singya da liwangi nyam do yo res.
And you might start to see that these two could not face each other. It's just too bulky.
Dila du dini do phusung chayra dini di chagi yo maris khaina na chigo sa thagi res mang thagi res di.
These bases are what spell out the genes in all organisms.
Dila du da di khari shiden chadi wala na rigze gi shiden chadi go res samala.
As I mentioned before, all our cells have the same DNA. My toe and my brain have the same chromosomes in them with that DNA.
Tanganzo thabung khande jita na di nang lo DNA sama khari la na chipa res ni go na ni thanda thabung jilem bai na re thana shi kangbe zugu ni thabung jilem bai na di nang lo DNA ta na chipa res chromosome chipa chadu res.
But cells that are in different tissues are very different from each other.
Hine thabung phungdup loga ta na sugu dinala chashinal phungdup loga ta du bala Quran na lo thabung minda cha de res.
So it wouldn't be so useful for a brain cell, a cell that's in my brain, to make muscle.
Tadi kechmu chai di pendobu yo mares lebegi tabung digi shane dubainas.
So if my brain cell made the protein that's in muscle, I would just have no brain. I would just be a muscle head.
Dila duda lebe tabung jura, the wangza tasu chawara. Di tandang azu shane gi tabung dugu yo na ni dinanglo la ni shane ma shi lebel hang maras.
In the same way, these are cells in our intestine.
Tadi tabung do cha cha na azu gyume nang gi tabung di res, gyume tabung di cha do res.
They have proteins in them that break down our food.
Ta dinjo tabung nang lo khari la na tigar ju se ma juye jura digi ma shige di di tigar di nang lo res gyume tabung.
Some of them secrete acid to make your food break into pieces.
An Quran su khashi gi khari sugor la na kyurze ri juya kyur di tungu res digi ani se dinjo ma shi du res.
You wouldn't want these proteins in your muscle chewing up your muscle.
Di kiranzo ngarazo ishane nan gugu yoro lan gugu yo maras di maya gyudura juye di chigi ras cheza di ngazo shane nan gugu yo mares.
So the big idea is that we have the same DNA everywhere, but which genes are used to make proteins specific for that part of you are different.
Dilo do DNA tawena tabung changma nam chikpa chadures ne subu gi chashi loga hindu kala wongbo loga chadidu ne khari la tigar khari gugu dulani dilo pabe ani rigze dinzo mindo chadu res lega chidandi.
So DNA is the blueprint for every possible protein your body can make.
Tangazo tigar duwe gi dubcha di ta cho di thanda shizaro di khari chadu lan ngazo rigze di chadu res.
But these proteins are what are actually doing the work in the cell.
Tangazo tabuna la chele dubne cho khari chadu lana tigar denzo res.
We have about 25,000 genes.
Tangarazuna chashawaina ani rigze di khazur lawaina nitinga dong sures.
And remember, we get one gene from one parent and one gene from another parent.
Ani tingu dwas rigze chi khane ni rawol na phani raw che chi mani rawi ras.
The variation of different variations in our genetic material within the population is what allows evolution to occur.
Dilo di ngarazo rigze na chogdu na taw na rigze mindo chadu ras dinam tis khebayo du gala digi ani ngarazo chogdu na lo mindo chune dan pelgir lo phengi res digi.
But DNA hides in the nucleus where it's safe on a chromosome.
Ngazo gi thanda chobong na lo DNA di khol ler lawaina tening na lo res.
Proteins are made out, do you know cytoplasm? If here's the cell, just any kind of cell, this is where all of our chromosomes are. But this is where proteins are being made.
Ta di thabung bacha shasra dinzo khari lana chobong res ani tigar dinzo khol duggur lana ne del duggur res chila tening gi chila.
And this is the nucleus. This is the word I'll use for it. And out here is the cytoplasm.
Di tening di res ra thabung gi chashe and di thasi di res.
So somehow this information from the DNA has to get out here where the proteins are being made.
Ta khari chugur lawana DNA na nesu di tigar susa del mal dugu ras nesu di.
So it sends a messenger. It says, you go, I'm staying here, you're my messenger, you go out to the cytoplasm.
Cheza nesu di ma dugu gola del ponya jitang gu ras ngorangi ponya jitang gu res nesu di yama kenge.
And that messenger is called RNA.
Ta nesu yama kenge jura ponya di khari chadur lana RNA chadur res.
Almost like DNA with a few differences.
Ta DNA RNA di DNA daw res phalam doj ne khebar ti res dinila.
So we should compare here and here. This is going to be the gene that's going to make a protein, and this is going to be its messenger.
Ta di ani rigze chadur res tigar dumni ge ta korangi ponya jura di ani RNA di ponya di chadur res.
All DNA has this base. Remember I said T's are going to bind with A's, but it sends its messenger. It's replaced, the T doesn't stay in the messenger. Something that looks a lot like it is a U, and that's what's on RNA.
Ta dini daw res lara nila DNA darni. Di DNA nal kharir lana ngajo je mata waina pisung nani yachik di T lani yoas mala. and RNA na la kharir lan tis chu gyo duni T sab la kharir lana U res.
And you see they've just colored that blue. Now we have a new color. So it was green, yellow, pink, and now it's green, yellow, blue. And that's very close. That's the RNA version of the T.
Dila duta dina gyo du gala di kharir lana kyaw roj dos and servo singya dos dina ta oina kyaw seo ani ngombu chai res di thanda ngajo DNA na la T yo di sab an di U di cha de res.
There's nothing magic or super important about that except when you see all these slides that are going to have how amino acids look, and there's going to be all these U's.
Dila do ni ngaran so ta amino cure jo dina ta du gala ani amino cure de khande me ta du gala dina lo khari thoa erlon thanda U chi dos di yo res amino cure na.
And don't think that a gene is this big or this big. Genes are going to be very long.
Ta kiran zo rigzel na diga sugir lana dende mares gene thanda rigzel la de rimbu shu yo res.
The alphabet for DNA is just 4 bases, 4 letters.
Yigita oina thanda ngajo DNA rang yigita oina di ducha dilta du gal khajo lana yigi shi dub de res.
So to get some kind of a complete sentence, it has to be very long because it's only working with 4 letters of the alphabet.
Dilo da gene rigze so du gal dende mangbu shu gi dub de res ra an thanda dina la dubcha khari cha de lana yigi shi di res.
Don't concern yourself with this top bit. This is the RNA that matches the gene from which it came. This is the messenger that went out.
Ta thanda di gang di DNA res ra and di da tu me thanda ngajo ne chu ma tangai RNA gyuni tangir la ra RNA ga ji di cha de res.
That's why I had to mention these U's. The original gene on DNA would be G, T, T, GCA, GCA, T, G, T, and so on. That's how the gene would look, but this is how its messenger looks.
The DNA na ta oina G che T, T, G, T, A, G, G, T lan thungu res ra. Ta di korang gi phonya di cha du wala ani di phonya di dende ju res. T sab yu le da di lawar.
You're going to learn a lot more about this next year. I know because my husband's talking about it to the third year class.
Ta di kor la thande ma shibta thanda ani khena cholorim shiwala ni sanda gi khena cholok di chi res.
So we have the RNA and they put an M in front because that means messenger. It's the messenger.
Ta di dudul di RNA res and di gala M chunju ji gya da di khala wala na messenger phonya la yu res.
And this is the beginning of a protein.
And di khari go chu go la na tigar go chu e di res.
The cell knows that when it sees a GUU on a messenger, it should put this amino acid right here.
Ta ngajo thabung na la thanda RNA GUU thoina and lamsa tigar khari shagir la na di gala amino cure di shagir res.
So on, when it sees a GCC, oh, that's not a GUU, it needs another amino acid here called alanine.
Ta thabung na la GCA thoina swa di thondu gala koran chu gilamsa ni ni shawa de chibo marshal ani koran pa be amino cure shemba ji shagir res.
So this is how the sentence gets spelled. Groups of 3 put in a given amino acid. 3 an amino acid, 3 an amino acid, and so on.
Dila da di chik group do ji shagir res di group dan di ani sum dig ne ani mama amino cure so ne di dig res.
Proteins are usually hundreds of amino acids long.
Ta thanda tigar la jora di ta oina ani amino cure ringlo khaga sur la na gal has te di group shagir res.
Don't worry too much about this, but I wanted you to see these are all the possibilities for these 4 base pairs. So all the combinations you could have of these 4 things are represented here.
Ta thanda yigi shi yuas dubcha so di shi ani tikdam manda ten de rest dina res.
And you can see in general the first 2 letters are super important. Because if you change one, here it's AUG is important. It makes a different amino acid than these sets of 3.
Yigi thanda di yigi sum gi dub shadas yigi ni di kechim bu chagi res thanda AU che ani G imbaina dela ani amino cure chik su res and di gang sum di ta ana AU chik par res ne yigi sumba di kha cha do dinzu gi ani ya amino cure minda loga ji su res.
The third spot we say has some wobble. In this case, valine, a certain amino acid, can come from any of the combination. The first 2 are important, GU, but the last one can be any of the 4 base pairs.
Ta thanda di amino cure shimba ji res valine la di res di ro cha sha na yigi thangbo ni di khechum res ra gu changman gu ji bares ta di wo de uca ji lo ga cha res ne dinzo digdan dende di wa na amino jur khande ji tugur la na di thanda valin di cha res.
Certain sets of 3 are very important.
Chesa dende sum digi da cha di khechum shu cha res.
When the cell sees on the message AUG, that means start here. Let's get going. Start my protein here. Start. Let's start up.
Ta thanda kisi RNA la AUG di thong ai na sra an khar chi la na da di khar tang la na thanda trigger so go chuk la da di tang res di.
In English, if I want to have the beginning of a sentence to make it simple for everyone to read, I would capitalize the first letter.
Ta inji na cha sha na ta di chigrup di khechum bu toy in bai na yigi thangbo de ani ngi yigi che tu res.
In some way, this AUG is the same thing. It catches the cell and says, start here.
Di la di thanda nganzu inji chigrup na pheje chho na shi di AUG di thapu na lega di ro chi res ko dine sugla ni dash ti res.
At the end of a sentence in English, we put a period.
Di la di ta chigrup di zo sa jala inji na cha sha na nganzu check di thi yos.
It helps our eye, it helps us to read, it helps us to see on a page how things are going.
Di la di khare finger la na nganzu shoguna ta wajang gi khande chigrup di khande image khagas sam sha go image ra di lo wajang ta wajang gi ha go res.
And I figured out yesterday in Tibetan, there's a period. It's something like this, right? Way down. What does it look like?
Pyu na la ge chi yo asa di sabla. She ge yo di yo ros.
So the cell has things that say stop. This is the end of the protein. Don't go any more far. Stop. And those are these 3. These sets, kick the bucket, sets of 3. UAA, UAG, UGA, UGG mean stop.
Tila duta zam shala da tingi jura di khari cha de na de la UAA, UAG, UGA landa. Di sumgi an khar da khari ti la na zam shala ngi da tingi di res.
What does an amino acid look like?
Amino jur dusta du khande ju dusa.
These are the amino acids that might come from our DNA.
Ta mara so thabu de DNA na ni amino jur tende roj cha de res ta inas.
This is the simplest one. They differ by what they put down under this carbon. This is the part, the side chain.
Dila duta sur la thanda igi tuen shada di ani dila side chain lagi res.
And they have very different properties or similar properties depending on what's here.
Dila du thanda sur la side chain la na ta suting lan disara dende roj yo res ra. And di cha sho na di ge ani thanda amino cure gi khejo mindo sugu res.
You can see this one, proline, is a circle down here. It's tied things together, and that usually makes a protein twist and take a bend.
Dila duta di proline ro cha sho na dela khar cha sla na kholo ro cha dos ko go. Di ge ani trigger de khuge da de phengi res.
This is the one that always starts the sentence. It has a sulfur in here. It's a bit unusual.
Ta di ro cha sho na dinala khare lana sulfur musi di res di minda ji cha de res ko.
Darwin said that all organisms have come from a common ancestor. This DNA is shared by all cells. DNA has proven itself over vast amounts of time to be the best way to pass on genetic material.
Dila duta Darwin gi khare she lana king khandi ji na changmo mebo chi grace lani she res. Ngara zo thabung khandi ji ta na changma na khare lana DNA yo res changma na chi tumong gi cha ji di cha de res. And down she on the reguching ni ma pe jura di any curing boot and the tower and some corridor lana gudeshing gusha tower and DNA the chad res.
Darwin didn't know anything about DNA or even genes. He was making his observations from a wooden boat going around the world before we knew any of this.
That and the Darwin Koran Chashawana and the Rigzela, the DNA, the decoration mares. In a Korangita, Sarnia Swiship and the
It was in my scientific lifetime that we learned all these amazing details about chromosomes, base pairs, DNA, RNA, protein. That's been just since I was a kid.
Ta thanda DNA da RNA la da tigar la da dinzo da drigze la dinzo sama khadu ni bochu la na ngara chungdi gopsan dinzo ne chu dinzo shares seri ba dinzo.
On the other hand, I'm really old.
Ta dil de enchi khari chade la na nga gengo chi chade res.
So, this stuff about now we have the technology to figure out DNA sequences to see what the genes are making in their proteins.
Ta tingsa churi yagi rodu gala ngara zoida thanda rigze la na khandi jume di sama ta tu res.
And with computers, it's easy to line up proteins to see how well they match from me to you to that tree to that dog, how our proteins are the same or not.
Thana shi computer na ya dig tigar sama leni ta mi gi tigar da simji shimba tigar dinzo leni khagas thungu do di sama ta res.
Rather than write out all the big names of the amino acids, they're just designated by a letter. M is methionine, V is valine, and so on. So each one of these different letters of the alphabet now is a different amino acid.
The amino cure ming rede tidana ringu shu chagyo res cheza an duni ta M do chashona amino cure methionine lan phugu res on V launa valine lan phugu res tinzu ming du chagyo res yigi chikna.
So this is the protein composition, the amino acid sequence of a very old protein called cytochrome B.
Ta thanda di tikar khari gajten dola na ningba ji chade res cytochrome B la di chadi gajten res.
It's even in bacteria, certain kinds of bacteria. It's in plants, it's in people.
And this cytochrome B the thasi bacteria na la ures ani shimba simje shimba na la ures.
If I had covered up this, what they are, I bet you could guess which ones were closest to each other and which was the most different organism.
Ta di ming denzo thanda simje ming tuen do thala. Di ka wana an keranzo denzo ta wa cham gen keranzo simje di da di nye shure sha di da di tharinga dulani keranzo she to ruas.
So you see the protein composition, thank you very much. The amino acid sequence of this protein in rice down here is very much the same like in all these animals.
Dilo de ta o di khari ga tuen la wana ngazo de ro de ga je amino cure ge tik sha gyo di tuen rest gardi. Gang denzo khar na simje shimba denzo res. Ta dine this minda che ya o la do this simje shimba da thung duas.
So in a way it's remarkable how much this protein looks like the human protein.
Dilo de ta thanda di tigar da ani mi tigar thung duas ha sa duas ti.
Yet if you look at animals, so forget the last line, animals have more amino acids that are identical. This whole part here, from here on down, is identical amongst all the animals listed here.
Ta simje pura rang na ta hoina di sama thungu dos amino cure di sama seuj ga gyo din sama thung wara di sama thun di dos.
Of these four animals listed, cow, chicken, horse, human, which one is kind of on its own, different?
Ta thanda di simje shi ming tuen dos thangbo di phaju sha do niwa di chade sha do sumba di tare sha shiwa di do mir do ta dinala denzo shi ta hoina kono zuna na minda o khandi dos ta.
Which one?
Minda o cheshu ta khandi na dos di shi ne lo su yo khandi do di shi khandi chicken.
So 3 are mammals, 1 is a bird.
che zanta kiranzo chade logar shela rasti chu chana ligi ras shimba sum dikhar lana nusu simje res.
You can see the chicken starts having some differences here where the mammals have still kept the same amino acids.
Ta thanda nusu simje ta oina di sama chikba thunde dos ina di thanda chade ledu wala de amino cure minda oji dos dina.
Chicken has an E and mammals all have an S.
Ta thanda di ro cha oina chade la khari lana E dos ani nusu simje sama S res amino cure ra di sama.
And with computers and with smart guys, they can really line up proteins and make conclusions about how closely related we are to other organisms.
Dila duta chele bakoran so computer natta ni di digro di dig sa jela that amino cure trigger di thungu dula dani kengu ni khagas ki nyelo me di koran so shetu res.
DNA can change. That's how we can become different from each other.
DNA la de judo res di kenge ni ngara so minda chhagu res.
And you probably think we said this 1,000 times, but those changes that give a species an advantage are those that are kept.
Dilo do the yo draw ina niri dilo kebe yo ina and dinyar digo res nyatsa chigo res.
So the changes, the mutations are random, but whether they're kept or not is selected for by nature.
Dilo do the tanda yo draw jora DNA gyurdo draw di dugo yo res nyeme res. Ina ta gyurdo chimba di tanda ganzo nya gugo yo nyatsa chigo yo chiyo mar lai di damye su cha de lana rangjung ham gi damgo yo res.
See, here's the N, here's the UAGs, here's all the stops in our amino acids.
Yelang anzo amino cure la kaldum chi langi jora stop langi di chigo chigyan kiran de la dos shedi.
I want you on this slide to forget this. This is a bit misleading.
This chi ta tagda maimba doji cha de res tanda di ten shegedi.
So cells have to divide. Bacterial cells divide every 20 minutes.
Ta tabung den zo kha ki yo ras kepel du yo res thana shi bacteria thasin bacteria kepel do di khagas du yo la na kama nishu na lan kepel re du yo res.
Each new cell needs a double-stranded DNA helix, a molecule of DNA with the two strands.
Ta tabung khande je na DNA khande yo la na chusum ni den di cha de res ko ba ni yo de.
So DNA has to divide and make a copy of itself. That's called replication.
DNA korang khare chi yo la na khacheni dashu chi yo res dila ani replication dashu cha di cha yo res.
So the DNA starts to unwind a little bit and molecules come along to make its matching pair, its mate, sort of.
Dila do ani khacheni an dudul de zule ni korang gi cha dig du go res.
So there's a new strand of DNA growing just as these are separating. As soon as it can separate, it's making a copy to take to the new cell. So this is the old strand on each, from this original molecule, and they're both going to go to separate cells now.
DNA khache sa yela ani korang gi dudul shimba de zo ani dashu shini suya go zu go res. Ta thanda dina ta oina ngombu di ani kuba nyingba di cha de res. An dashu shu sawa de di res.
So in this cartoon, green is always with purple, red is always with yellow.
Ta di rimuna ta oina jangbu di khari nyam do cha de la na khadu na moment di nyam do res. Ani mao di ani seu nyam do yo res.
And the idea is that the next molecule going to the other cell, to the new cell, will be exactly like it was here.
Dila duta di dashu chisa ra di thabung na la dashu chisa ejala di dudul di khande jor la oina ngone thangbo khari yo di nang shin ju res.
And in this side, for this original strand, the molecules that are putting things together got it right.
Dila duta dina la dudul de zo digdu kala takta digu gu res.
It made the new purple, the old purple got a new green, the old red got a new yellow, all the way through.
Dila duta takta digu la na yeni moment yo sa dela jangu yora thanda sawa sujo dina la jangu yo res thana shi mao nyam do sawa khari cha de na seu ji res thana shi jangu nyam do moment de rang ro di res.
And when a cell divides, it has to do this for millions of these pairs.
Dilo duta sa mangbu cha de res di cha de.
So once in a while, a mistake happens. On this one, this is the original DNA.
Ta de DNA ngoma jora ngone thangbo di imba cha sha nas ngombu di.
But the machinery to make its right pair goofed up. And here's the green doesn't get a purple, it gets a red.
Ne kabre kharchi gorla na dita thuje do la ga chi res di ani nundu shor ni kharchi gorla na jangu nyam do moment ni ba nam ju imba na di mai ba che jangu moment sabla ni mau nyam do thu res.
So the daughter cell, if this was the mother cell, the two daughters that are happening, one's going to get an exact copy of what the mother cell had, and one's going to have something different.
Ta di thabung amad na cha sha nasra dini ne ani ni di khol dur na chung thabung na du res dina ta du gal chigla khari la na ni ne ama thabung khari ba de chibu chigya res an shemba di khari la na nundu shuni tis mi do cha de res.
It doesn't happen often, but when you think it has to go and read over millions of bases, mutations occur.
Dila du di suya di khazu du la na sai mangbu jor la ra piso di suya du gal ani kabre jurdo du res di gen kheba cha res.
This is shown here, where the original gene started out as a G and it went to a C.
Ta di do mba na jurdo thanda cha sha na thangbo ngoi la khari la na G res jela jurdo to ni di sabla C le res.
Now you might be thinking, she said the third one's not so important. Maybe this won't make a difference in what amino acid comes into the protein.
The Kiranzo Yeni Galaisha Nashi Yeni Sumba de Digas Ketchumbu Maris Tigar Duvela and Chesan Kebashira Sugu Marisamgi Gires.
Certain environmental things make mutations occur more often.
Dila do Gurdo di Kabri Karulana Kuryugi can get any Gurdo Manga Yungures.
For example, ultraviolet light from the sun can cause changes in base pairs, especially I think at the T's, as I recall.
And Tanda Muchi do Yora Uni, the little Chashana, Angazu di Pusungi Chachi Yachik and the Gurdo Dread in the Kenchi Yures.
That's why sometimes for light-skinned people, the sun can cause cancers because it's changed the properties of a cell right there to grow too fast.
Dila duta shamdo kara rig juda dinde indugala nye thanda muchi yura dinde rigani di khare pakpe gi thanda khejula gyuwa tane ani cancer ne ba inde chaya kangi thigi res.
I wanted you to, these slides are good to simplify things, but I don't want you to think that this much DNA would make a protein.
Ta kiran zol chik shuya yins ta digas do cha shawana dinde gi ani tigar surulan dishi gi mares.
But mutations can occur in this gene, or in this gene, or in between genes where no protein is being made.
Ta thanda jurdo dre jora de rang durulan dishi gi mares. De drosire de la drosire yana dikila di tigar ke dugu mares dinang lagi jurdo drosires.
And again, it might have no effect, good effect, or bad effect, where bad effect is defined as limiting your success as an organism.
Ta jurdo chimba la jora digi ani shugi te sigres shugi hom meba yo sigre thana shi shugi ya te sigres shugi duga te sigres.
So, remember, I think that one example said it was ACC going to an ACG. Huh? That's what the mutation was going to be. It still puts in the same amino acid.
Ta mo la shio na shi thambo amino cure ACC bas jurdo chine ACG charla roas dende roj yo droina kheba room maras khais na diniga itanda amino cure disum da puran sum khari ta cha derla na tigar chikba cha deres.
There's no question this is a mutation, but we call it a silent mutation. It doesn't change the protein's amino acids.
Di jurdo revela na res amino cure guro dro res ra ine digi tigar la kheba masu dugala ani silent mutation la lagi res dela.
So, mutations lead to variation. And that's what makes changes in a population, which is what evolution is about.
Jurdo dro kengi ani mindra chaya jora na so badi chawi res digi kengi en thanda ngajo ani phejur chudu res soduna.
For these mutations to change to make a new species, which I think Megan's going to talk about, takes a long time.
Ta jurdo chine ani thanda nirik sawa ki re loga chai le jora di chayla ani thujo ringu shu borgi res di ge Megan gi di korchi res chala.
This is the gene ATG led to an amino acid. Here became AUG. That's to the cell. Start now. Here's the methionine goes in there. This is the beginning of our protein.
Ta di gang di DNA res ra and di har surla na RNA sugura di dashuchi ani RNA digi thanda di cha shawana AUG res amino cure and this so member na zo khari ragorla na methionine go chugla di da di res.
So remember, the T was in the gene, the message gets a U. ATG in the message becomes AUG, which becomes the amino acid methionine.
Ta thanda di gang la cha shawana di do TAC di chabla wala khari chagorla na AUG chagyo res RNA dubde.
And these are all different amino acids joining up to make this protein.
And this samata amino cure chig gel tu ruda di tune ani jela tigar di dubu res di wa di tigar da.
And the cell knows to stop because this is the period. This is stop. Don't go any farther.
Ani koran so thabungi khadu ha gur lawana digas la jugdom chiguya khailana di UGA la di jugdom chiguya da di tigar res.
Although the slide has tried to show lots of amino acids, most proteins are larger to get more amino acids than this.
Dile dita thanda dil tigar chikten dos tigar phecho khari lawana tigar dile che res cheza amino cure gi thanda thema ro dite ona dile ringa chawi res tigar shimbana.
So they've made a hypothetical, a model of maybe this protein looks like this.
The thegas koran jo cheba chine tar tigar lai dita thanda digi tigar ji dubai na dinde ji dubure lan ten sha res rimuna.
And that shape is a large part of allowing it to do its job in the cell.
Ta tigar gi korangi thanda yeb jura digi ani thabu na chele khari dugu me di kheba shu su res.
But let's say there's a mutation. Then instead of getting the amino acid that used to be here, we get a new one, something different.
Dila ta di ro cha sha na di maus ten dos di gyurdo chi oting res. Ta dinde gyurdo chin du gala ta dela ngazu gi amino cure mindaj ra res.
That change can really change the shape or the ability of that protein to do its work.
Dilo di gyurdo tro kingi ta ngazu trigger ji du ras di yeb dia manda che ani korangi chele loga ji chi res thabona.
Some amino acids like to be around membranes in fat. Some amino acids like to be where there's more water in the cell, and those amino acids give the protein its form and its function.
Dilo de tigar khashe khari lagaw law na chuna dinge khashe khari lagaw na shasi ri lagaw res dil ta be ani quran sugi ta yeb da namba din su chadu res.
I used this slide before, but this is the same idea. These are the 3 base pairs from the message, well, from the gene, they skipped the message, that are going to make a protein that looks like that. So this gene is going to make a protein that looks like that.
Ta di amino cure che din zu ra amino cure che res ani rigze dinde do jiyo aina digi tigar dinde stu res lan pesun sha res dina.
This gene has a G here, but it's at the 3rd position. If this gene had GCA, this one has GCG.
Ta rigze re wa di rigze res ta di mi thangbo ji do di na la GCA res kora rigze na ta na di rigze na ta di GCG res e gi chap la G le de res.
But it puts the exact same amino acid right here in the middle of the protein.
Ta di chik jo gyo ina ge ina amino cure la ta na chibo ji kyang res.
So the protein ends up looking exactly the same and does the job just as well from either gene.
Chezanda rigze khari imbai nai thanda tigar jela dugura chipoji kyang dug de res an chezani rigze la kheba thanda di kheba doy ne ge thanda tigar la kheba doy mares ramja res.
In this case though, the gene went from AGA to AAA.
Ta di ro cha sha na thangbo gene di khari yorla na AGA res. The dinal ta na AAA cha res.
That middle spot is more important for the message to be read correctly.
Ta thanda nesu di loela di ki di khechimbu cha de res. Nesu di ta loya jila.
And it ends up, instead of being this protein, the amino acid called arginine, it's something else called lysine.
Cheza di gyo drinking gi ele ro chabla. Ani di thanda amino cure arginine chabla lysine cha res.
And now the protein has a different shape and may not work so well.
Ta amino cure di gyo dro so jela ani tigar gi kheba du res ni. An di lega ni ni khari chaw khari du gyo di ta ta du du yo mares tigar sa di.
This, where its amino acid just got changed, nothing got taken out or added, is called a point mutation. Just one point is different.
Ta thanda di do jin bai na di igi chigrang chi gyo dro res ra ji gi sab ele ra di la an zo point mutation la gyo res check gurdo rolan di sa dela.
So this will lose something, I think, in the translation, but this is a sentence in English that makes sense. The cat saw the dog. That makes sense.
Ta thanda nga zo inji igi logo yo las di do cha de res thanda di igi logan di ta nga zo inji na cha sha na shimi gi ki thong res la jora di do cha yo res di la du ko ji dos.
But if I just changed a letter here or a letter here, I would still have a sentence, but it wouldn't make such good sense. This would say, the hat saw the dog, or the cat saw the fog.
Ta dene ro ju sena sa gyo ta na igi chi gyo ta ra ta di head la na shamu la ra shamu ki thong re la dele cha yo res an ta na shi the cat saw the fog la na mu ba da siwa ro la ra di shimi gi ani mu ba thong ras la di cha yo res che gyo dro ina.
But I would still, my eye, that would look like a sentence.
Sorry?
My eye would still see that as a sentence.
Tangarang imig di tadu gala dingi do do ji thong yo res.
If I just lost this letter, think of it as losing an amino acid, then this is gone and it goes the blah, 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 and that's what this is. The C is gone and now everything looks goofy.
Ta thanda di igi lo dan shi da sum ding ni lo ra di lo du gala khwa dos shimi ki thongarla ni this sigrup dikdugal ta thana shi kesi di igi chik si jigdo di matyo minus di tena da igi sama ya chik chik shi dugra dila di sigrup di khome jo chadu res.
Likewise, if I stuck in something here, now I've got something equally that doesn't make any sense.
Thana shi dene ni ngoi ni chik tuen dugal sigrup di khome jo cha res ra thana shi chik nembe gyawa lage ya sigrup di khome jo cha res.
By losing a letter or inserting a letter, I've shifted the whole relationship.
Dila duda igi chik ma luwa ina res ma themba ina res digi ani tanda sigrup da igi chik dewa jo chama tu sha res dina.
And those are called frame shift mutations.
And di tanda jurdo dila frame shift mutation lagi res.
So, are mutations good or bad or nothing?
Ta tanda jurdo dra del ni yur thida di ani yabu re na duja re na kheba yo ma res nan di thiwa di res.
I think commonly we call them a mutation if they're really bad.
Ta chida maran so idam ju jurdo la di khol pheju chir la na du yesa du cha do jil cha shagi res.
If they're not bad, we call them almost alleles, like blue eyes, brown eyes. I don't think you would think a blue-eyed person has a disease.
Ta ngjo thanda chidang namju ke sha dil allele la ni shagi res. Mik mombu ge allele du den la ni shagi res shayad isidi.
Since talking to you last, I looked up about blue eyes. It came from the Baltic Sea, which is up there by Sweden, and about 10,000 years ago was the first blue-eyed person.
Ta thanda Arctic ne deola ni nyama thangbo chunge dis Sweden you said right? Sweden la di ne gula ni mik mombu chunge thangbo dela cha res sa dela.
His or her parents must have thought that was a mutation.
Ta mik mombu thangbo chungdu wala phama ikharisam borla na ta di nazar ro indo sam res manda ch thoira.
Now it's in about 10% of people around the world, so we think of it as an allele.
Ta tingsa ta du gala thanda zamling gi kya ta wa nas member dela kya chu zamla mik mombu cha de res dila du ta ngajo di kyo ro chi yo ma res di alil na sha yo res.
These are things most of us would call mutations. There is a mutation that's inherited, obviously, that gives the person an extra finger.
Dila du thanda jurdo chi kharila na phaman phudul ma dre judong che yo adende chagyo res di jurdo di ani khari chagyo la na mi laba del zugu de ta na zugu thu res zugu chik thewa cha de res.
When some people, when they see a baby with that, they might like to have the surgery to take it off when they're little, but this guy grew up with it.
Ta mang che thanda jurdo di yudu gala zugu thuk chagyo ra del nga do dinga ni chik shagjo na ni ma me asu res ine dit phudi chasha na Quran zugu shares che zan zugu thu res kon.
There's a disease we're going to talk about, I think, tomorrow called malaria.
Ni naza ji US malaria di naza di korsangi she his.
It's spread by the mosquitoes who are infected with a very small parasite.
Dila di naza di khani gyuni yungur la na tugdang gyuni yungur res tugdang dila ni thanda shinden solcha yores Quran lola di gyuni naza di li yores.
Parasites have very complicated life cycles. They change their forms depending on when they're in the mosquito, the malaria disease looks a certain way, the organisms that cause it, and when it's in people, it looks even different.
Dila di nanglo thanda tugdang di nanglo shinden solcha yores di naza suyer lara Quran gita di ta wainas shinden solcha khandi yor la na manda ni yores ta thanda mi supuna yudu kala number minda yores thana shi di tugdang na yor kapsu Quran number minda shtim yores di solcha di.
For this parasite to cycle and keep living, it has to go from the mosquito to mammals. Mosquito to mammals.
Ta Quran soye che dura tanda shinden solcha shdo di khar chi yor la na tugdang na do na tugdang ni minale ya mini tugdang na do di Quran soy mise jungor dende Quran soy che jungor dende ro cha yores.
It's a very severe disease and kills maybe 1 million people every year.
Andi naza de ta nyenga ro cha yores khaina na logic na la misayat says me osugures na malaria digi.
And my doctor before I came here gave me my anti-malaria pills to take.
Dilo de ta ngara de gyagal ma yong khon la ni member gi ngala thanda malaria ko ay gi me dite sons.
What my doctor didn't know is that I have a certain kind of blood called O.
Ani nge doctor gi thanda nge member gi khari ha ku yo mar la na nge thana di o di yo ha ku yo mar res kurangi.
We all have red blood cells in us, all of us, that look identical under a microscope.
ta ngaron zo changma I thang na ta na thangma thabung yores an thadong cheshin na ta ni changma chipo thung res.
But on their surfaces are certain kinds of proteins that make different kinds, we call them blood types.
Dilo du ta thagi rig la ni ngazo yawa chi yores di khandis chi yor la na thagi thanda ngol la ta na thabung di dilo an tigar mindo cha yores.
Some of these blood types, it could be A, B, a combination of both, or O.
Ta di rig mindo dilo ming ta yores khashila A legres khashila B legres khashi di ni ga de A B yores khashi A O yores.
People who have O, even if they get infected, don't get so sick.
Dilo du thagi rig kisi tha O imbai nas rig de ani malaria thanda di tukdan di so jab thanda naza di shiras nu yo mares.
And in certain places where there's lots of malaria, the disease is selecting for O people, for O blood type.
Dilo du ta damga chedu gal koran so khari damgi urla na tha O rig judo adinde rig de damgi ures.
And we'll talk more about, well, we can talk today. If a malaria is in your blood cell, here's a good blood cell, here's a healthy blood cell. Here's a blood cell that has the parasite, here's one that does not.
Ta dela thama thabung ni cha shawai nas chik dela khari urla na thanda shinde so cha di yores parasite di chik dela yo mares thangbu res.
This infected one can cause the healthy ones to clump and stop blood flow.
Dilo du ta di naza phogyo dirwa di ga ni thangbu yo dil nyam du naza di chini nyam du thui ni ani tha den ju chik jare jora klam cha yores.
And if you have O blood type, it doesn't do that so badly. So you might be infected, but the disease is not so horrible.
Dilo du ta thanda thagri O imbai na thanda chik tham di jare jora di nyunga res di gi keng gi khana nyenga chayore la na chayore sine naza di shiras kangi shira tigyo mares ne badila.
This poor fellow, 5 generations ago, his grandmother had a mutation that caused her to have hair like this. I thought he had painted his face black when I was looking at the slides at home. This is hair.
Ta di seu thong mendolas di khar la na pures di sama ta tombal puki da di rig res korangi mirab nga khong la da tanda gyurdo di chini di shugen di ta tanda koran thoya res dongla puki da di res.
This is a very rare condition. They can count how many people in the world have ever had it.
Ta dende rig jido dende rig de tanda ngara zo member na ta na kombo shu cha de res cheza thanga ge yo dende cha de res di kangi di.
This man lives in Mexico.
Tanda mi jido koran desa hor la na Mexico res.
For 5 generations now, his family has had children that look like him. Not all, but some.
Ta tanda koran mirab nga cha de ra mirab nga ringal tharan kangi di the de res France no mila.
And somehow in his grandmother, if this is the X chromosome, I'll draw it as a double so that it looks like an X. On one part of her X chromosome, a new piece of DNA jumped in here and made it just a little bit longer than it should have been. It has a little bit extra DNA than normal.
Ta thanda di Quran so momo di DNA ta na khale charla na namju DNA X digas pa dio ras rindo digala DNA gi chashe chik digal thewa jaro che ani chachik ringa chai res.
So this is extra DNA that is not where it should be.
Ngola yo mares kugu yo mares di ani DNA thewa di cha dio res.
This is a dominant mutation.
Dila du thanda ta wangyur chinge do la ora dinde cha dio res di gyurdo di.
So that means just her chromosome, her other X chromosome could be just perfect, but just needed on one, and that makes the disease.
Dila du ta chromosome thanda chobong den cha di yo ras di ya chi yo ra di thangbu imbai na kesi di yo aina di gi wangyur ni di naza di kangi thi yo res.
This fellow was treated very badly in his village in Mexico. The whole family has had, of course, troubles for generations.
Ta korangi thanda longba jura dela ani korang zo nami thama kangi shu thi yo res chi so na ta dan dubja ta yo res.
That he's a devil or a werewolf.
Ta du do chi yo res du da de la ora da dinde roji ani changi rig ji do dinde rig do chi yo res.
So to make a living for his family, he joined the circus.
Chezang koran nangmi chi du ngo di thabdun che circus le jo din an su res.
But over time, he missed his family, they missed him, so he went back to his village.
Ani tanda tabdun chisa dina lega chirdinya nangmi shu tinya rangja kura thongsi la phalo res.
He's still alive. It's been very hard for him, but his family and other families who have this kind of trouble have been now studied by scientists.
The Quran chen tang shi Quran nangmi cheni dinde tanda pu dongbal pu mangbu king rijora dinde riglani tanda seriba denjogi taje shimju chigi res.
And by looking at the DNA of the family, that's how they figured out that everyone who looks like this has this extra piece of DNA here.
The dinde rigla seriba taje chigi lara taje chidu Quran so DNA na kharinya or lawena DNA di thewa jido dinju rigna la Quran so DNA thewa yo ni res seriba.
Maybe this is not so. Maybe this is ahead, do just tell me. But this man is XY, right? So he got his X chromosome from his mother.
The tanda di na poro po insan korangi tsubung tana XY chagiroas. And the X ni legyo insan X di khane ni chung orla na mani chung shagires.
He got his Y chromosome from his father.
Ani tsubung Y di koran pa ni chung res.
So when he makes sperm to have children, some will have his Y chromosome and some will have his X chromosome.
The korangi tanda kham kau di dugdugala khashi nala khariorla na thabung khashi nala X res khashi nala Y res.
There's nothing the matter with his Y chromosome. This is perfect.
Ta tanda kesi kham kau khashi na Y yorla ora dinde rikta oina di thangbu res an kangi khebu jiyo mares kila.
But all of his sperm that have X chromosome will have this disease.
Hina da korangi tsubung X ingi rijora dinde yimba na di naza di khede res.
So let's say he has a little boy.
Ta pesha oinas ra koran la puji ke oinas.
Will he look like his father or will he have normal skin on his face?
Ta puji chung oinas ra ta kora pala do thanda pu mangbu yu yungure yana thangbu ju yungures di thiorish.
What do you think?
Kyan ju samju baldus pala do yungure yung mare. Okay, some of them say he won't look like his father.
So here's the new baby. The baby is a boy. He got this from his father, which is perfect. I guess I should tell you the mother is perfect too. The mother doesn't have the disease. So this chromosome is good. This chromosome is good. It's normal baby.
Le ama ya neba mares ra an ama neba mendukal X di thangbu res an Y di khane chunglan di phani chungres an phadi Y di thangbu ra chezam po dil naza di phom mares.
Okay, you'll learn more about this. But let's say he sent his X sperm into the egg. This one is terrible. And this is a little girl.
Ta pomuji chung ainas ra ta korangi X di li ras ta di nazar phogyo di chadu res.
This is the bad
chezam pomudil kangi thik res na pu ke di.
All you need is one chromosome to be bad. All his daughters will look like that.
Dila du chubung chik la nudisho ainas ra ta da me ainas un koran pomo hajuk ke aina kangi dirang thedi go res nazar di go.
If you could select the gender of your baby, you could wipe out this disease in this family.
Dila du ta sosugi pomo dame yo ainas ra bu ani di migyu korana thanda nazar di me asetu roas.
He has a big family in Mexico, and his daughters have it, and then his nephews and so on have it from his sister, and so on.
Korangi aja da pomo da chamala dide nazar ngi chadu res korazo nangmil ta ainas.
So these are just things we would call mutations. This one's not so bad. This one's good if you're an O, and this one is pretty horrible.
Dige di shiras kangi mendoas di imbaina yaga ras ola kyudo jora di pengi ras the di imbaina yagu mendoas.
It's horrible in a way that you have to function in society. It doesn't kill him. He still can have offspring, but it's what I think we would all think is not a good mutation.
Dilo du tang azu chizona dingi do di kangi thiyo ras chizona ta dang dubja du nazainde she kangi shu thiyo marte kangi ta chizona kangi thiyo res dilo du di jurdo de ngaran so imbaina yagu sing maras.
It doesn't affect his fitness, and his sons are perfectly healthy.
Korangi choden da dinde kala kangi yo mares tigi.
This is a very rare condition. When I saw the slide, I didn't even know what it was. I had to look it up, but it is a well-studied phenomenon.
Di ta tanda thugyon rojosh to di rig di yo res and kombu shu res ngara thangbo ta di kharim ba yabu seuji hago masons ine di thwa yo res di kangi di. 콤보지 차도 레스디
What they think happened is this extra DNA makes a gene up here next to it not function correctly.
딜레도 탄다 디나라 DNA 태워주라 디갈 아니 델라 릭제주레스 딜레가 야고 치기요 마레스.
And as is often the case, something that starts out as a scientific question becomes a money question.
The Terigi to a chadir, the ineta, do the pendulum laura, win the deer to a chadir, the dish chadir resting some.
So once scientists figured out what's causing this, drug companies are trying to use that to prevent baldness.
Diladuta, Pesha Suegi Tablam Rose Chair Lara, Chikarishi Urlana, Seribaita di Karigi King Yungudula, Naza di Taja Shidugola, Anita that song that deer, Koranso Karchigurlana, the meat that Chamaking Rigida, the Dilla mean swear. Dita manga kigura, di tapshi chigyo res, di tsongna piju che tapshi chigyo res kronzoi.
Which may be why they're finding out so much, is because the drug companies are giving money to study it.
Dila duani chigda khari cha dula no koranzo suene suye res, tanda taji che di sama ngui di jo men suene ta tsongle chingi jora din zuri gi tigyo res sora din zuri.
Some of these kinds of diseases that are so rare are called orphan diseases.
Ta di naza din jo kumbu shu yu bala naza din zuri la orphan naza lan lagyo res.
Sometimes it's only if it affects too few people, then no one has the money to study it.
Kan di mi khashij mashil yo marwas kumbu shu yu bala ta din jo meo chana ani ngui tangwa lage taji chisa yo mares.
So we have almost a half hour, 20 minutes or so. How about some questions?
That chujik the common issues doshas and tanda tiwa chilen khashij chinas.
I think this DNA, RNA, amino acid, protein stuff is overwhelming, and you'll get it again next year in more detail. But don't forget it between now and next year.
The DNA, RNA, tigar la de this mount hajo roji chag do asine ta digan shifta lorim shiwal le du kheran su jangu resra. Ine thandal lab shajo de themba je yo mares.
Okay. Questions?
Tiwa US.
DNA, RNA, and ETSI.
Okay, remove ilam nityon rochidas. So he wants illustrations of DNA, RNA, gene and protein through diagram. He wants to see these diagrams.
He wants to see what?
Diagrams of
Should we
DNA,
go back to the
RNA.
slide, you mean, or?
Ani di karani ni slide na tengyo dramja teng gasa.
Okay.
Chi da lawre. So is there gene in isolation from DNA, or is there a specific part in a DNA which we call gene?
Yes, absolutely, absolutely. So if this part of our DNA goes on and on and on, there might be one part here that we can say for sure is a gene, because we can look at its base pairs and say this is going to be a protein that we all can easily see is going to be a protein.
The DNA bacha shastra di na bushthensa the DNA res ta ne dineni janguni thensa dini de ani rigzere lagi res khaisna dineni di ne dineni tigar dubu res cheza manju di ne dila rigzelan lagi res.
This DNA then makes the message, which is called messenger RNA, and that involves a migration. If this was the nucleus where all our chromosomes were paired up, this involves the RNA has to go from where it's made off the DNA and has to get out to the cytoplasm. So all the DNA is hanging out in here, except for mitochondria, and all the RNA is moving out to the cell.
The DNA ne di ani rigzere la ra di ne dene ni khare dugur la na nesu di sugu res nesu la ding anzo nesu king khare cha de la na RNA di cha de res phonya di messenger RNA m ge gyo jo di mRNA di cha de res di thanda anzo thabung na ta na khandir la na di tening yos tening na lo la DNA res the DNA dinin khare suere RNA suera RNA di ma thaji na res.
Once RNA is out here, it needs a lot of other molecules to bring all those amino acids to it. It's a very pretty it's a very pretty process, but it's going to be for your next year.
The RNA the Tazinali raw, the Lesa Jala Korangi Kajilana, and the amino cure sama killing trigger to bed the chigures. The Jurim Trotan the pay now ruris, the Canazoni, Lorim Gemma del Changres.
This is a double-stranded molecule that has all kinds of proteins protecting it in the nucleus.
The DNA Dudul Chusum Yidin the rest. The call the Sungyo Chinisharlana, Tening Kimo Yora, Kimo Yo Tening the Nala Sungyo Chininya de rest.
This messenger RNA is a single-stranded molecule.
The RNA Ponya Jura, the Nezuking messenger RNA, DNA Chusu the Kuba Chig resti, Kande rest.
It's subject to degradation.
The DNA said that the DNA can't to rest RNA.
It'll be here for a short time maybe and then gone, to make the protein and then gone.
Koranzo Karchinglam to this Shiringal rest, Tigar Dub Aniam Meachadu rest.
Which allows the cell to be responsive to what genes it needs right then.
Koranzo Karil Yalin Tigur Lawana, Tatanda Manzo Tigar Karidu Buguerlani Rizajora Dile Yalin Chingish Chadu rest.
Once a message makes a protein, that protein may live for a long time.
The Denini Tanda Nizu Digi Tigar Dubsara, Tigar di Durumbuni rest.
Let's say in my brain I just learned to play piano. I learned it in my fingers, but I hope it's in my brain.
Chasha and Natangi Lebanon Tanazo Rocha Piano Jua, the Jaya Shimainas.
The DNA in my brain has not divided since I was born, probably.
The Lebanon DNA Jura, DNA and the Chaguchi Rolana Chaguchi Maris Nakirinichi Chashawainas.
But it may have made messages since I've been born that allow me to learn things.
Digi Karisur Lana DNA di Nisu Suejura, di Janeda di Samadi Nisu Dubires.
That's kind of a crummy example, but that's the idea.
The Dipisu Kenzo Rojichaso Sine Samsu Taran Restodani.
The RNA allows the cell to be nimble and to respond to what the cell needs.
The RNA di Tanda Tabula Kari Gugu Majora, Dilapa Beni Korangi Yalinchi res.
The protein that it makes may be very stable.
The Tanda Koran Tembushira Yomarla in it, Korangi Dubi Tigar Jura di Tembu res.
The RNA may go away.
The di Dubsa Jala RNA di Meachadu Ross Toshi res.
Then the cell maybe have some new insult or a new environment, or this protein maybe got lost. The mRNA can be made again because the DNA is over there and protected.
The Tabun di Tanda Koyu Shimbajnali and the Tigar di Meachainas, and DNA Gikardu RNA Dubu res.
So the short lifetime of a message makes the cell responsive.
You don't want
that message around forever, making a protein that you don't need anymore because the protein is still here.
So this RNA is made by DNA or has it come from outside?
No. It's actually so pretty because it's the perfect match of the gene. Go back to the double helix picture. So if this is all DNA, the RNA will almost be like the opposite strand. I mean, it'll be just like the gene that it's making a protein for. It'll be just like this, except the T's and A's are replacing.
The DNA RNA kubachik lembaina RNA dindro jures. The hari kebar yorlana DNA nangla T yoros, T sabla RNA nangla U yores.
Every base on that message matches the base on the gene exactly if there's been no goof ups.
The case in Roma shoina dig nanglo khari yo di chikbachikya RNA nang yores.
Except the U is a T becomes a U.
The dichibu kheba yores T sub U le jora di kheba yores.
RNA koram kugi dedik nang zotinchi koram shilon zotinchi nang washing.
So he's saying again like Is RNA made inside the nucleus or does it come from outside?
Yes, it's made out in the nucleus. The protein is made in the cytoplasm. That's why it's the messenger. It's taking the DNA's message out to the cell.
RNA la di thanda ngazugi ponya la odi ching la gores swe di ko sugur la na tening na sugores and di tigar swe la ni mathazi na dugores.
There are beautiful pictures. Well, they'll show you next year. There are beautiful pictures of proteins being made, amino acid being added from a certain special kind of microscope.
Dila do ta thadon she mixe chingi dende pheju chini ani amino gyur den chigi gel thuni ani tigar dube jora di ani longe indu sweres di kenan zoni lojema tungures.
This was all figured out just when I was a kid. This is not like Charles Darwin science.
Dila do ta nga chunju yumbe kapsu di ne su di sheres and Charles Darwin dende thuzu go do yomares di seride.
The pieces to make RNA, the A's and the C's and the G's are floating around in the cell by themselves. And then a protein comes and reads the DNA and takes the right pieces of RNA and puts them together in the right order to copy the gene.
Tadanda tabung na la tanda A, U, C, G lash do. The tabung na yo res wrong and tigar gi dos lene ani DNA la desing DNA and RNA sugu res.
Right, those bases are used for other things, not just making DNA. You may have heard of a molecule called ATP, which is how we store energy. The A is the same A. There's a GTP, same G.
The tigar chik khari la na ATP la di res, the A la di chik ba cha di res tanda nga zo DNA na yo di.
These bases are used for lots of things, but they're really used in the DNA.
Tanda phusung denzo shto denzo kechimbu cha di res, khaw ga sala phejo yungi ji cha di res. Tanda dina cha sha na DNA na li res.
What we're talking about going from DNA to RNA to protein is what scientists call the central dogma, because it's very important to biology.
Tanda ngonzo DNA ni RNA ni ani tigar doya jora dila ngonzo theri by central dogma la res.
And we often use an analogy to explain it to students. We talk about DNA being like all the recipes to make your favorite foods from your family and your ancestors, and we store them in a cookbook.
Dila dita DNA la di nesu nyangi ro thep ta ngonzo khala ro sugu na khari sugu me nesu sangma dina yoro as di ro ngara zo ita khandes chagu me nangmi khandes thongure ta dang da nesu sangma di sangma nesu di khol nya de la na DNA na nya de res.
You keep that book in a special place to keep it safe. Our cells keep it in the nucleus.
The thep ding ngonzo kechimbu insan ani yabu chini nya ro asunggyop chini di ro ngonzo gi thanda DNA ding ngara zo thabung na la nya de res.
When you want to make one of your recipes, say for momos, somebody reads the book and copies the recipe down onto a piece of paper.
Tadi nganzo khala sudan gi thep ro di cha de ras dila de da momo sugu na momo khande sugu me di nganzo ma shu ro as thep shobo jinala.
You can take that piece of paper from your safe place, the nucleus, and take it out to the kitchen, our cytoplasm, where you're going to make your food.
Tadi momo suya sudan shobo jira thep ni ma te ni dele ni khol ki ru la na thabjang na ki ru ras thabjang na momo su ru as nganzo.
Once in the kitchen, somebody else can read the recipe and make your food. Or read the RNA and make the protein.
Ta thep di thabjang na ki ru de shobo di thabjang na ki ru ras momo sudan de. Di swinge su cha de la na mi shemba ju res di ge ani momo su ge res di ngaro zo thabong na lo tigar du pi di ro cha de res.
That's good.
It's good to have a partner.
Di chi du lega chi du ro ba na ya bu dos.
Are there more questions?
Ani tyo shimba US wala chi gera. Yeah, one more question from there.
DNA que pas juste c'est ça t'as pris les bases c'est ça ni tabu les non le tene super monde au camp de ils sont souvent le tene pas juste 근데 매직 유리 차이가 있나 DNA 사야 뭐 같다 되어서 ils sont souvent le tene tabu gibi 나오니 pas juste 다 뒤에 나오니 DNA 이런 기가 아니 양의 DNA도 양의 super 이런 기가 super 이런 기가 ताकि कम बात की चाह में ना मुकम्मल चाह लो ऐसे बोलता यहाँ ना कि लोग ने जीएन मोड़ो थे तो ये सुबह कम बात की चाह दे ऐसे होता है खाली से
Ale tanda naza dindu yungi mirik jura. Dina cha shawana tabung chigran chigna le yore na tabung chasang le yore slan bitiora. Tabung kazu yo changmana la DNA thagrang subung thagrang yorwes. So his question is about the rare condition, the hair.
Yeah.
So he said there is an extra DNA, right?
Little
So
piece.
are you talking about extra DNA in a single cell or in all cells of that
It's
organism?
it's in all his cells, yeah, because he passes it on, yes. It's in his sperm. It's everywhere.
So in all cells.
But it's only, you can see his face is really affected. The gene is most expressed up in this part of the body.
Tadi tabung keje shana tabung changmana yores kurangi.
It's in all his cells.
Theodi, DNA theodi.
We're ignoring, for this DNA, RNA, protein, all the other molecules that contribute to this process happening. But this is the basic idea. This isn't the last time you'll hear about it, and you'll hear all the other processes like ribosomes and transfer RNAs and other kinds of proteins that are needed to make all this happen.
Tatanda durdi roji shesha restina. Tatanda diga la tigar sudu gala thandi rabota sugro da ani thanda keden chingi RNA da di changma jelo kheran di kor changres.
But for now, this is plenty to try to soak in.
Hine thanda thande char la kheran su digas ki shingba ina digi res nesu.
Thanda DNA da shu che ba da ne kana DNA ne tu ma te ne RNA ma ko ne tigar zo da. Ndo zo da RNA tigar zo ta mu sa ba je tu ra da.
Ale tabung sawaji dugi yor res ra tiora. Tigar su. So tabung sawaji dub rewe tiora. So when you said a DNA makes RNA, copies RNA, and then RNA makes protein, does it mean that when you say it makes protein, is it making a new cell?
No, your cell needs different things at different times. So it's not always, it's just making its protein product that allows, maybe it makes that cell now into a bone cell, or it makes that cell now into a liver cell. Certain genes have to open up, make their message, make their protein, and make specific proteins for that cell to become what it needs to be doing. It doesn't necessarily mean dividing. In fact, when it's dividing, it's not making new RNA, the cell is just trying to copy DNA.
Dila du thanda ngazugi tigar saw dubre la jura dila du thabung saw du shigyo mares dila du tigar saw du tigar digita ngazu ne khol me chashe khari la laga khari chigu me chele dil pa ben tigar digi khari sugu me di ta sugu res kisi rube thabung dubu na khora rube thabung la dubu res di rize di sene tene chigyo res thabung saw dubu re la ne dubu mares di.
In fact,
danda goe jura ta kebel choru khanagi chago choru shi da dinde chigyo mares.
In fact, it's sort of a generalization that a cell has to not be dividing and kind of taking a break to make the proteins that are specific to make it a certain kind of cell.
Dila du ta thabung kora ri khari sugu gu me jura di soela an thanda chele di drup de dinde chigyo res.
In an adult, hardly any of your cells are dividing anymore.
Sorry.
In an adult, not many cells are dividing.
Ta nganzo mi gemba ri chasha oina thabung mangche nganzo chagu chigyo mares.
In fact, the cells that are dividing are the cells where you meet the outside world.
Dila du thabung chagu chigyo gabso kheran da khori saw ji theo do chadu res.
So your skin cells are always dividing and sloughing off the old skin cells because they could be damaged by DNA.
Kais na ta nganzo pakbe thabung ro chasha oina di ani DNA den zo khadu na ta negyo ro shor di ge ani thabung ningba den zo rig di khachi gorla na la du res.
In your lungs, your cells in your lungs are meeting the air from the outside world.
Thana chi ani ngazo loi gi thabung ro chasha oina chi da deo chigyo la ora di ani ngazo lung ling yo ras khoryu ni.
And those cells are dividing to produce fresh cells lining your lung.
Dila du di khoryu nyam do deo chi di yor mashi thanda di thabung de ani khoran chagu chi yor lan di shigyo mares.
In your intestine, the cells are meeting your food, and everything that you're taking in by mouth is meeting your cells in your gut.
Thana sta ngazo yuma da dinde chasha ona se sagyo di chi ita yonga dos chadura di chi da ni te rescue me thabung de zo.
So those cells are dividing to provide fresh cells because there's a lot of interaction and with the bacteria and so on.
The di thabung rig de zo an thabung saw dubni anju se da din de rig ni ani bacteria thasin bacteria da din de thedu gala ni thabung saw dubya da din de chungu res.
But most of my cells are not dividing at all.
Ine ngaran ro cha sha ne nge thabung di chagu shu chi yo ma res. Chi me da cha chi bo res.
In fact, brain cells are probably they're very hard to grow more brain cells.
Cha sha na da lebe gi thabung do yim bai nasra ongse thasu din zura din de ani chagu shira chi yo ma res.
If you cut a nerve, you may have seen people like that then the nerve cell cannot grow back if it's in a certain part of the spinal cord. If it does start to grow back, it's very slow.
Ta thanda ongse thasu jik che wai nasra ta kangi dabgen ro sho ni che wai nas di ani kyar du sugura subuna di suya de le tuzuring bu shu gorgo res.
There's a lot of research now in science to take cells that are very what we call very simple, very undifferentiated.
Tanganju thanda cheriba de ju khari gal ta je chiwir la na thabung tab deuri de ju gal ta je chiwir res.
And make them into a special kind of cell.
Di thabung tab deu din ju kuran ju khari le gyur ju gorla na ta mixe thabung mixe rig jora den le gyur gu res.
So they're trying to take, if someone's had a damaged heart, they're trying to grow him a new set of good heart muscle cells.
Dila du ta ning ning kange yo enas ning na zo yo enas ani thanda thabung din ju gi ani ning gi thanda shani thabung rig jora den de dube gi ta je den chi yo res.
This is just the edge of science now for trying to take cells and make them into special cells to replace things that have failed.
Dila du thabung tab deu chigni ani thanda mixe thabung thanda chasha du gal ning gi thabung lara dinde dube ta je chi ta suya gi thapshi chi yo res.
Maybe very good, maybe a little scary.
Ta ya bu yu si re yang ang da di kangi yu si re sti.
You want to comment?
Ling jumsha chache ka mabuyo labe cha lapa kan chabon ro chile ban ti na ya ni mi pe na mi ta yana chasan tuya ni kaza reject yore.
Okay, so for example, if we take a single cell from this part of the body, can we find all the genes in that single cell?
But they're not all the proteins that DNA could make.
Ta kerangi ra thabung di lemba inas rigje chasang yore la ra rigje chasang DNA chasang yore rigje di chasang yore sine tigar duggu yore la na tigar chasang duggu yo mares di thabung di.
Zishin ro yo ra zishin ro ni chabon na ya so pe na mi ki ko de duel kaza ti de.
So in plants, do plants have the ability to have the genes to make human eyes?
No.
Yo mares.
We're quite different than plants.
Kailan chishing the mi but loa cha de ras in the yo mares rigzedi.
But in my finger, we say when a gene is making its protein product, it's being expressed.
Dila du ta ngazu zugu ro cha sha enas rigzedi ge ne tigar su du la du gala ngazu gene digi la chiwa da express chi du la ni shi yo res te shi du.
So different cells express different genes.
Dila du en thabung re re ge ani rigze minda loga ani koran jo ta se wala ora di loga cha de res.
That allows that cell to become the kind of cell it's supposed to be.
Dila du thabung khari cha gugu me jora di ani rigze di khari se gu me dila ra lu res.
It's like you go grocery shopping, everything you want is there, but you just pick this one and this one and this one because you're making a certain recipe at home. Those are the proteins that are being made in cells in my finger, which are different from those that are being made in my eye or in my heart.
Cha shawa na anjo tabjang nan ji khala ji su gugu nas ta chongal rodu gal chala sangma yores ine khari goe ta khari su gugu na dil pa be chala di damgi roas di ro cha de res.
But that's why DNA is so great. It's stable. The same DNA is in every cell all over.
Cheza DNA le di tembu shu jores thabung khandi jina ta wana changma chi pa res.
Anda cheza pe mi juya de reject de mazo labe ki chabon na yores la di zang ni cheza mazo pe na zishin tu tina mi juya de yo ma se la do ni por khata ri shu yi zang 그래서 답변은 아주 뜻있게 아니 직제를 좋아지는 다다 무슨 직단 되어라. 아니 뭐 좋아지는 남식 있는 가서 미 주에 직지 뭐야 좋아 예식이 됐어.
미기 주메 토니 요 타에 요레스라
그래서 무슨 좋아지는 미
ता थंदा चिशिंग की ठापुंग जुरा दिल यो तांग नि की ठापुंग जुबे रिजे स्वे यो रेसरा so the genes for to make eyes in human, can we make changes in the plant's DNA so that it can make human eye?
Oh, that would be maybe over the difference between us and plants is millions and millions and millions and millions of years. So that's a big, you can't just change a gene and get eyes. There are plants that respond to the sun. I mean, in some way they know where light is and will grow towards the sun, but that's not, we couldn't put eyeballs in plants, no. Not I couldn't.
Ngai bo ene chidu samaris chishing da mi ru cha shauna saya mangbu I kheba sho sa resra dila dugola rigje dil kheba shu do sa res an thanda rigje di gyo ta ni ngaju gi da mixwe mi gi rigje di dina yo res la na yung mares ngara shewa na yung mares.
When we say that the DNA is the same in all cells, we mean within one organism, all of my cells have the same DNA. All of your cells have the same DNA.
Which are not the same.
But my DNA is not exactly the same as your DNA.
Ta thanda thabung kingwe chig na la thabung na lo rigje sangma thanda DNA sangma chik pa re la na ta ngaran cha sha na ngi thabung cha za na DNA chik pa cha de res ya mire ngure ta na chera ba re la ni su su thabung thanda kingwe chig la ta du gol khorang gi DNA sangma chik pa cha la go res but
that is why you can take just a small part of a root from a plant and grow a whole new plant with leaves and flowers and everything just from that small part of the root cells. Some animals can do the same thing. There are some flatworms where if you cut off a lot of their body, they can grow it back. If you cut off their tail, the cells from their head have that DNA and they can grow a new tail. We can't do that, perhaps because we're too complicated. We cannot make plants grow eyes because it is not just one gene needed to grow eyes. It is many, many genes that you need to grow eyes. Scientists can now take a gene from one organism and put it in a different one. We call these genetically modified organisms. But we are not yet able to take many, many genes from one organism and put it into another. And I think we're over time for tea.
Yeah, just a few minutes.
Tea break.
Oh, that's such a person.